Good morning, everybody. Just for those of you who don't know me, probably the majority of you, so I come from Stellenbosch University. I'm a microbiologist, and we specialize in soil microbiology. And I'm really interested to see what the effect of human practices are on our soil microbiomes. And by doing that, we've moved into agriculture. So most of the work that we do currently is in the agricultural space, particularly regenerative agriculture, and mostly on wheat and maize. But through our soil health service, we've now started also receiving quite a lot of potato samples, and I'll share some of that with you. About rotation systems, and I think we need to start with the definition. So what is soil health? And I think it depends very much on who you ask. It's actually it's a loaded term, I think. It's defined as the continued capacity of the soil to function. So we like to refer to it rather than soil functionality, rather than soil health. So for those of you who don't know, if you look at your soil and you take about a gram of soil, you'll find about 10 trillion bacterial cells there. We're talking big numbers today. Fungi, about 30% of your biomass. And you can actually use these organisms. They can fix nitrogen for you. They mineralize most of the nutrients. And in agricultural systems, most of your nutrient cycling goes through this microbiome. So I think it's really important to understand how they work and how they react to what you do in the field. So what are these functions? They have many, many, many functions. But what is really, really important to understand is that microorganisms do not work as individuals. They work as groups and mostly in consortia of multiple species. And what they do is really context dependent. It depends on what the nutrient status of the soil is. It depends on who else is there and what they are doing and which plants you are planting. We can link productivity to microbiomes, so we also know that if you have a really productive soil, you have good plants, your plants look healthy, it's most likely that you have a good microbiome. But if you have problems, definitely you have a problematic microbiome. So please remember that. So you can influence this microbiome and you can do various things, but if you really want to move your microbiome and shift it, you have to play around with pH, water availability, and then, of course, the type of plant that you plant. So the space that we work is right here, at the surface on the plant root, or the rhizosphere zone, because that's really where things take place. So if you want to determine what your microbiome looks like and what it does, and is it functional, this is where you need to investigate it. So in the zone, Okay, we have a lot of beneficial and pathogenic microorganisms, and that's where they function. And that is where you would like to have an impact if you do anything to encourage these microorganisms. So the other thing that's really important to understand is, while these slides are trying to there we go, is that either these organisms can have direct and indirect functions. So direct functions, it fixes nitrogen, it produces hormones, it produces all kinds of things, nutrient solubilization and mineralization. But it also have indirect functions. And you can read through that yourself. I'm just worried I won't get through this. But very important, it helps protect your crop against pathogens. So if you have a healthy soil microbiome, you can actually balance out the pathogens. So they may be present, and I'll show you some of our results later, but they won't have such a big impact. So two of the terms that we very much use to define them is resilience and redundancy. And if you get that and take that home today, that's really important. So when we measure our microbiomes, we don't look at individual species. We look at the resilience of the total community and the redundancy. And with redundancy, we mean we need to have one or more species that can perform the same function. So the more restricted your microbiome, the less redundant it is. So this is just a sample or just an illustration of work that we've done. So each of these little dots represents a single soil sample, and the colors represents different crops. And the first thing that you would notice is that your crop selects its own microbiome. And that you can't get away from. And we have seen this across multiple systems, that if you have a wheat microbiome, it stays a wheat microbiome. The core microorganisms remains the same. And it's the ability of each plant to select those specific microorganisms that it evolved to work with. So, in regenerative agriculture, how do we manipulate, how do we increase our microbiomes and build a little bit of redundancy in there? So if you monocrop, and if you can see at the bottom here, that's your microbiome. Now in crop rotations, what happens is you have your crop, goes off the field in a couple of seconds, you plant your cover crops, or your crop, come on. There we go. So you take it off, and plant a new one. This is going to take all morning. Oh, there we go. And it's planted. Maybe it's just a reflection of what happens in real life. And this particular plant, so it's a different plant, selects a different microbiome. So now you have a mixture, and what most people do that are really successful in this is they leave the roots alive from the previous plants. So now you have additional microorganisms in the soil. They interact with the old microbiome. Some of it is shared, some of it is not shared. So now you plant your new crop and you have a much larger group of organisms to select from. So you're building this redundancy. So should you have water scarcity, if there is a bit of a temperature shock, it can now recruit other organisms from this mix or larger microbiome to perform the necessary functions. Now, if you intercrop, it gets even better. So now this microbiome gets larger and larger. And then if we really see the impact of regenerative agriculture is when you start introducing animals. And one of the big advantages of animals is they bring manure to the field. And of course, manure, there we go. Manure adds microorganisms to the field, as well as nutrients, but most important, there we go, carbon, nitrogen, and phosphorus, but also dead microorganisms. So we talk about the necromass. The necromass is a really important nutrient source for other microorganisms. They feed off these, and the more dead bugs you have in the field, the better. So this is basically what we are aiming for. So I'm sure this one is not going to run, so let's see. So in the system, what happens? No, this is not going to go. Can you just... We've got photosynthesis, and we know that about 40% of our photosynthetic products goes into your roots. And these products is excreted as sugars into the environment, and that is what attracts your microbiome. And there's specific receptors on your microbial cells that detects these things, and then they will gather around the microbiome. So this is really important to understand. So carbon goes out, attracts your microbiome, but we're adding some, in the next little bit, we're going to add some nutrient or fertilizer to the fields. And once you start adding fertilizer, you are actually breaking a really important thing, which is that nutrient limitation that drives the nutrient cycles. So there's a specific law, Liebig's law in microbial ecology or in any ecology, that tells you that the performance is only as good as the nutrient that it's in the lowest concentration. And we like that to be nitrogen. So if nitrogen is in low concentration, most of your other cycles are ticking over nicely. If you break that nitrogen limitation, your carbon cycle goes into overdrive. So you start burning through carbon and you run out of carbon in your soils. And the guys who run through that carbon is your microorganisms. So you're building biomass in your soil, but not necessarily a good biomass. So biomass, microbial biomass, can compete with your plants for nutrients. And they do sometimes do this very effectively. Something else we see if we have too much nitrogen is something like mycorrhiza will actually climb out of your roots and become competitors for your plants. If you have too much nitrogen, your rhizobia will not take on your soil. So now they compete for nitrogen and phosphorus mostly, and you end up with a system that only releases CO2 in the atmosphere and you need to keep adding nutrients. Now, if you can reduce the nitrogen input or the fertilizer input, and we've seen this very effectively done in the Western Cape, you'll also notice that it attracts this microbiome, and you start, if there's a nitrogen deficiency, immediately the group of bacteria that fixes nitrogen will kick off. So they have two types of hive styles. They can be saprophytic, or they fix nitrogen. And they only fix nitrogen when there's a nitrogen limitation, because it's an expensive process for a bacteria. And like everybody, they're kind of lazy. They just do what they need to do, not expend too much energy on things they don't need. Hierdie gaan my oud maak voor my tyd. Daar gaan ek grys hare hê teen die einde van die praatjie. Okay, so you have nitrogen. If you have nitrogen limitation, mycorrhizae will also go in. They're really excellent in mineralizing your phosphorus. And they also have compounds that is really hard to break down by other microorganisms, and that is where you start sequestering carbon in your soils. So these things are really good to have. You can measure, you can look, you can check if you have these organisms. So carbon sequestration is really good, and in theory you should have a good system. I think this is the only time that I'm using animations. However, if you can measure this stuff, you can manage it. So that's where the problem comes in. How do you measure this? And now I'm really happy to tell you that in microbiology in the last couple of years we've seen massive advances in technology which now allows us to study this microbiome. However, to our shock and dismay, we've also figured out that we know less than about 20% of what occurs in soils. The rest of them we have not seen, we haven't looked at, we didn't even know they existed. But we're working with that. So I'm going to give you a sample, just an example of maize that we've done in the Free State, just to show you how we measure this. So this is, if you remember yesterday, we saw some Shannon and Simpson's diversity index. This is a biodiversity index. Red is always your conventional farms. The other three are regenerative farms. And the first thing you see, your bacterial load or diversity is much lower in your conventional farms. Because the farmers introduce different plants in the regen farms, they have more bacterial species. Fungi tend to not be too worried. As long as they have food to eat, it doesn't matter who's there as long as they have something to eat. And their functions are very similar. We're very interested in the nitrogen cycle. And the nitrogen cycle, the microbes involved in the nitrogen cycle are very specific microbes. And they drive this whole cycle. What we have seen, is I'm going to show you some examples, we've measured the number of gene copies of the different enzymes in the previous diagram. So there's we'll just go back, so the ones circled in red, these are important enzymes that drives the cycle that's excreted by the bacteria and fungi. And if you look at those enzymes, yellow is the conventional farms, you'll see that in all cases, there are more bacteria in your regenerative farms than there are in conventional farms, always. What we have seen, and you see that with the MOA, which is that one, this is the one that tells you whether your biological denitrification system works. It gets completely switched off when you add too much nitrogen to your field. So your microbes are no longer fixing nitrogen, they're no longer mineralizing this nitrogen, they just simply sit there and they become very lazy. So, just to end off, this is an example of potato samples that came through our service. And this is done with high-throughput sequence analysis, which basically means we've taken out all of the DNA in that soil sample and we sequenced it so we can have a list of names. Now, you probably can't see the list of names, it's very small. Those are just the 50 most important ones. So it is 50 out of 1,400 species that occurred in that particular soil sample. There are many organisms. What is really encouraging is that this bottom red and orange part are mostly bacillus. And we see this across the board in our soil samples, as we have a lot of bacillus species there. We see an increase in regenerative farms. So these are two different cultivars. You'll see there are slight differences. And we do see that, slight differences. What we've also seen is we could pick up some of the pathogens that are in the top 50. Once we have these sequences, we have algorithms, we put them into boxes, bins, and the bins tells us what their function is. So it looks at what they potentially can do based on what we know about them. So again, you can see that the two different cultivars differ very much in terms of the functions that they perform. So this one on this side is much better in carbon cycling and nitrogen cycling than the second sample. Incidentally, the second sample also had a lot of scab in it. So pathogens tend to also decrease your function. For the soil fungi, there's about 170, 180 species, so much fewer species, but they are bigger than bacteria, so that's fine. You can identify plant pathogens, saprotrophs, and a whole list of other organisms just by looking at the sequencing. So, and just to make sure that the functions are actually functioning, we do enzyme studies, so we put them in through a system where we look if the enzymes are actually working, so we look at the whole community. And these are the two samples. Carbon was not too bad, but you'll see that one sample, the beta-glucosidase, which tells us if the carbon cycle is working, isn't working so great. So now the farmer can go back and check and see, can I do anything? And there's a couple of things that you can do. You can add more complex carbons to the soil, maybe compost. There's a number of things you can do. The acid and alkaline phosphatases tells us if the phosphorus cycle is working. And it would be either or, depending on the pH of your soils. And then urease tells us if that nitrogen cycle is ticking over. So we can learn a lot by just looking at your soil microorganisms, but I have to warn you, there's no ideal. So I have many people asking me, which microorganisms do I need in my soil? The question is, it doesn't really matter as long as they do what they're supposed to do, and the plants will attract their own. So there's no ideal community. Soils are spatially and temporally heterogeneous. One size does not fit all. Your microorganisms will depend on the context. We find that a potato microbiome is actually a little bit more restricted than a wheat or a maize microbiome, which is really interesting. When you test biology in your field, test more than one metric. Just one is not going to tell you everything. And please remember that, like beauty, soil health is in the eye of the beholder. And with that, I would like to thank you. And I'd be happy to answer any questions during the break.